Hello, everyone. My name is Chi-Song, and I am currently a postdoctoral associate at Computational Biology Department from Carnegie Mellon University. Today, I am going to present a software package that has been recently published in Genome Biology. So this software package can cluster the genes from the pseudo-time ordered single-cell RNA-seq data. To begin with, I would like to show you this slide, which shows the workflow of the regular single-cell RNA-seq analysis. So what you usually can do is that you can build this gene expression matrix where the rows represent the cells and the columns represent the genes from the raw sequencing data. And once with this expression matrix, you can reduce the expression matrix into the lower dimensional embeddings by various algorithms. And then based on the lower dimensional embeddings, you can build clustering of the cells and identify different cell populations and perform differential expression between the different cell populations. So further, you can also do the trajectory analysis and identify the lineage relationships between the different cell populations and estimate the velocities for different cell populations. And this basically will show you the directions of the progressions of the different cells. So people usually call these as cell fates because this basically shows how the cells can change along the developmental trajectories. But what about the gene fates? This is relatively less investigated, right? So to fill this gap, we propose this framework to identify the gene fates from the single cell data. So here we start out again from the gene expression matrix. Then we build the trajectory tree using the common trajectory inference tools such as Monaco3 or Slingshot or Pega. This basically can give you a trajectory tree kind of structure. And then based on this information, we can select a certain path or a branch from the trajectory tree. Then we map the cells back to this trajectory tree and select a certain path to extract the expression patterns and aggregate the gene expressions and extract the expression patterns from the certain path. And based on this information, we can further perform the gene clustering using an algorithm called scSTEM. And this has been applied for bulk RNA-seq data in the past 10 years and which has been really successful for the time series analysis. And based on the success of the scSTEM algorithm, we can further apply it to the single cell data and cluster the genes using this algorithm. So scSTEM algorithm usually works with pre-generating the gene profiles, then assign the gene expression patterns to the different gene pre-generated gene profiles by calculating the distances between these genes to the profiles. So I'm showing you one example here, and this is the output from our clustering framework. And on the top, these are the cells from natural killer cell set. And we cluster the genes based on the trajectory information extracted from natural killer cells. And the end results can be visualized. And you can see there are red clusters and green clusters, which shows the activated genes and the suppressed genes. So furthermore, our framework can also identify the clusters on different branches, then compare these clusters across the different branches. We do this by performing hypergeometric tests to identify the gene clusters with similar genes across the different branches. So it will be interesting to investigate these green clusters and the red clusters because they have similar genes, but they are showing different expression patterns across the different branches. So we also perform benchmark testing for the different methods, including our methods. And the results have shown that our method can identify these natural killer cell related genes better than other methods. And the results also have shown that our running time is pretty efficient as compared to other algorithms. So usually our tool can produce the results within 1 minute given more than 100K cells. Finally, our tool is fully available as a Shiny app-based GUI tool, and it requires almost a zero coding experience to perform all the analysis. So if you want to look for more information, please refer to our GitHub repository. And this is the end of my presentation. Thank you for your attention. Bye.